Yet another Mendo Life seminar. Today we have the pleasure to host N. Dekas. I hope I pronounced it right. She is an assistant professor at Stanford University in Earth System Science, studying marine microbiology and biogeochemistry. She is interested in how microbial life affects the chemistry and climate of the planet today and throughout the time. Her research combines tools from molecular biology and isotope geochemistry to identify and quantify microbial metabolic capabilities, activity, and interactions with a focus on single-cell imaging approach. Currently, she is a Simons Foundation Early Career Investigator in Marine Microbial Ecology and Evolution. And prior to joining Stanford, she was a Lawrence Postdoctoral Fellow at Lawrence Livermore National Lab. So with this as a short introduction, I want to hand it over to Anne to tell us all about just single cells in marine environments.
Thank you so much. It's really a pleasure to be here virtually with all of you today. And I think this is one of the silver linings of COVID times, is being able to connect virtually with a lot more people than we were in the habit of doing before. So I hope that this is one of the ways that we can continue some of the things we've learned during COVID. So I will be talking today about primarily single cell approaches to look at microbial activity, primarily in the marine environment. Here we go. So as Neha mentioned, I run a deep sea microbiology and biogeochemistry group at Stanford. You can take a look at some of my fantastic graduate students and postdocs here. We study a real range of systems, primarily in marine environments, but also in some terrestrial environments, like hot springs and industrial salt flats. So today I'm going to be talking about a few specific stories kind of from our menu. But our overall goal is to understand the role of bacteria and archaea in biogeochemical cycles, the past, the present, and the future. I'm motivated to do this for a few reasons. One is a storytelling approach to explain the climactic events in the history of the Earth. Another is to search for life on other planets. So one of my primary funding sources is NASA, and we're interested in understanding how these extremophiles could inform our searches for life on other planets, and to predict future changes in Earth's climate. And this is probably the one that's caught my attention primarily in the last few years, to understand the feedbacks between microbiology and climate. So the specific questions that we tend to ask are who does what in situ, so which species are mediating which metabolisms, what influences who does what and how much, so getting at the controls, like the mechanisms behind what dictates the rates of activity, and will these metabolisms act as that positive or negative feedback to climate change, so we can actually predict what will happen in the future. So one of the big challenges in asking these kinds of questions is just the spatial scales that we have to bridge. So we have several orders of magnitude from individual biomolecules to the size of an entire planet that we have to kind of piece together. And each one of these different kind of places on this scale requires different methods. And I'll be talking a lot about the methods that show us the single cell range in this part of the scale. Can you see my pointer when I'm circling it?
Yeah.
Fantastic. These single cell techniques, so we can understand not just how the communities work in bulk, but how the individuals of the community work together to create that bulk signal that we see from a bulk analysis. So for those of you that don't think about the marine microbiome all the time, I thought I would start out with a introductory slide. So about 30% of all of the cells in the oceans are in the top about 150 meters where light penetrates. So here we have light and oxygen. About 55% of the cells, which is the majority of marine microbes, are in the dark, still oxygenated waters. And in the top 10 centimeters of marine sediments, we have a whopping 15% of the total marine microbiome, which is a really high percentage given what a thin volume this is. And we have extremely high densities of cells in these surface sediments. So in each one of these layers, we have very key metabolisms that are both producing and consuming greenhouse gases. And we care about understanding the mediators and the rates and the controls, one, because of this greenhouse gas production and consumption, and two, because of the overall biological carbon pump, which is sucking CO2 from the atmosphere and eventually burying it in the sediments, but it has to get through kind of all of these hurdles on its way. So today I'm going to be talking about three very specific stories, actually one from each of those three realms I just mentioned: benthic microbial activity in the dark ocean, and then finally diatom fungal parasitism in the photic zone. To give you an overview of how we collect our samples and perform these activity assays, I'll start out with just two slides that kind of summarize this for the entire presentation. So we use Niskin bottles and sediment cores. You can see a couple pictures of how we do that here. We also use manned submarines and remotely operated submarines for some of our more specific sample collection. And to get at the question of activity, we primarily use stable isotope probing. So I'm going to explain this kind of in detail here so that we're all on the same page for the rest of the talk. This is a cartoon of a mixed microbial community. And if you just plucked a couple cells out of this community, they would have a very predictable ratio of 15N to 14N, same with 13C to 12C, and several other elements that have stable isotopes. What this allows us to do, since this is such a predictable natural abundance ratio, is add substrates with the less abundant stable isotope as a label to see if they're capable of consuming this substrate in their metabolic capabilities. So here I have, as an example, I have 15N2 to see if there are nitrogen fixers in this bottle. If there are, that 15N ends up incorporated in the biomass, and the ratio of that cell ends up higher than the natural abundance. So this is kind of a very fundamental concept of everything that I'm going to be showing. We can measure the isotopic composition of these cells several different ways. One is isotope ratio mass spectrometry, which gives us a bulk measure of the entire community. You need a pretty large scoop to get this measurement. You can also do density gradient-based DNA or RNA-SIP, where you extract the DNA from these cells, put it through a density gradient where you actually physically separate the DNA that has more of the heavier isotope in it, and then sequence all of the density fractions so you can identify who was incorporating that heavier label. This is not one of the techniques I'm going to be talking about today, but is certainly a great approach for certain questions. And finally, nanoscale secondary ion mass spectrometry, or nanoSIMS, which I will be talking about quite a bit today. The differences between this method and some of the previous ones is that it's actually a single cell measurement. So you're getting an isotopic composition of an individual cell. It's quantitative. You have a spatial analysis, so you can see the difference between cells that are connected versus separate. And you can link function with phylogeny and get a particular identification of cells if you couple it with other techniques upstream of this analysis. So there's a lot of options here. So let's start with the first story, the benthic microbial activity in space and time. So why do we care about microbes in marine sediments? I've already mentioned that they're particularly dense, one of the densest microbial habitats in the marine environment. They play an important role in marine and global biogeochemistry because what they consume and produce there does interact with the water above. It also is important because this is kind of the last hurdle in carbon burial. So as I mentioned before, with the biological carbon pump and the ultimate fixation of CO2 in the atmosphere and traveling down to the sediments, if it can be permanently buried in sediments and ultimately subducted, this is kind of finally how we get rid of CO2 in the atmosphere. But any of the microbes that can respire that fixed CO2 in the water column or the sediments end up just converting the CO2 right back up. So we care about this fraction of the fixed carbon that is buried because it ultimately controls concentrations of oxygen in the atmosphere and the redox of the surface of the planet. So it's pretty important. Previous approaches to quantifying benthic microbial activity have really focused on catabolic approaches. So what I mean by that is metabolisms that harness energy. So particular ones like sulfate reduction or aerobic heterotrophy. And while that's really useful to know, each one of those looks at the activity of a particular group, the particular organisms that mediate that particular metabolism. So it's not really a whole perspective quantification. Approaches that look at anabolism have the potential to quantify all microbial activity because it's agnostic to the particular catabolism that the organism is mediating to harness energy. So examples of that are looking at concentrations of DNA and RNA, cell counts, but these methods typically aren't as quantitative and they're harder to roll into biogeochemical models. So stable isotope tracer experiments, as I mentioned, can be a quantitative way to look at total activity, but there have been very few of these conducted, especially in deep sea sediments. So that's the approach that we took. We added 15N labeled ammonium. Ammonium is a substrate that almost all organisms are capable of assimilating. So it is this kind of universal substrate that can help us get an idea of the total anabolic activity. Yeah, so that is what we used. So we collected samples off of both the west and east coast of the United States, so therefore in the Pacific and Atlantic Oceans. Our samples in the Pacific were along a transect that varied 100 to 4,500 meters water depth, and our samples in the Atlantic, slightly smaller range, 300 to 1,400 meters water depth. And at each of these sites, we collected cores that looked a little like this, if you can see it, and divided them into 5-centimeter intervals. So we could also see resolution with depth. And here I have a little cartoon in the background. It's essentially what this Pacific Coast transect looked like. We had much more resolution along the slope and then a few samples out in the abyssal plain. So along this gradient, we had over an order of magnitude change in pressure, large changes in temperature, oxygen, and organic carbon. So we're really seeing how a lot of different parameters could be affecting activity. So we did over 400 stable isotope bottle incubations. Here you can see the process where we go from the cores to the sections to the bottles. Two fantastic graduate students who led this work are Bennett Capilli and Nicolette Meyer. And I particularly want to call out Nicolette because she has led the analysis of all of this data. So in addition to our 15N ammonium proxy for total anabolic activity, we also added 15N2 as a proxy for nitrogen fixation or diazotrophy. We added 13C bicarbonate to quantify autotrophy, and we measured 13C glucose as a rough tracer of heterotrophy. We could get into more detail about why this is not the best tracer for heterotrophy and why actually subtracting this measure of autotrophy from the total is better, but we did it both ways. I'll show you why that was necessary in a few slides. So we did look at who was there. We extracted the DNA and RNA and are now looking at the metagenomes and metatranscriptomes. I'm not going to talk about that today, but I did want to show you just a little bit of 16S data to give you an idea of who is in these samples. So this is from our most shallow Pacific Ocean site and our most deep Pacific Ocean site. You can see that the more shallow samples were dominated by Delta proteobacteria. That's what's in pink here. And that was actually consistent out all the way to about 3,500 meters water depth, mostly Delta proteobacteria. But in our very deepest samples, we see more of a diversity and particularly an uptick of chloroflexi and planktomycetes. So when we look at the total anabolic activity, which we quantified by the uptake of 15N ammonium, you can see the shore is over here. So you can see that there's a distance kind of decay function where there's less activity the further you go from the coast. Similarly, if you look at how activity changed with depth at each of these sites, so this is the depth integrated rate, you can see that activity generally decreased with depth into the sediment. So in terms of what's controlling that, we did wonder if it could be nitrogen limitation. So in our previous work, we've seen more and more examples of nitrogen limitation in marine sediments. That's not something that people typically think about in marine sediments, because when you have decaying organic matter, you often have the production of ammonium. And so you have increasing ammonium concentrations with depth. But in surface sediments, you often don't have that much ammonium. And we've seen a lot of examples, particularly in places of increased carbon loading, where you have nitrogen limitation and even nitrogen fixation. And we've seen that primarily at methane seeps and whale falls, like I said, where you have a big input of additional carbon. And we were wondering how kind of widespread that was in regular marine sediments. And one of the reasons we sampled where we did was because we wanted to see just regular marine sediments. As funny as it is, that's actually one of the sites or the types of sediments that are really undersampled in the deep sea. You often have cruises that focus on kind of exciting methane seeps, hydrothermal vents, weird areas, but we don't have as good of a sense of what's going on just in the regular background sediment. That's of course representative of most of the ocean and therefore really relevant for biogeochemical cycles. So we looked at pore water concentrations of ammonium, and we saw that in many of our samples, these concentrations are less than 25 micromolar. The reason that concentration is significant is that's the concentration at which diazotrophs in the laboratory are inhibited by ammonium. So below this concentration, you might see nitrogen fixation or organisms that are converting N2 gas into ammonium for nitrogen. We also looked for NIFH genes, and we did find pretty extensive NIFH, at least amplification. We haven't sequenced all of this yet. I'll show you in the next slide. So here we're looking at the pore water concentrations of ammonium in blue at all of our sites with depth. These are all of the sites at which the concentrations are below 25 micromolar, so which might host nitrogen fixation. You can see particularly in the deeper samples, you have low concentrations of nitrogen, and particularly at the surface. And in this sample, we did actually sequence the NIFH genes. So if all of the samples that we amplified, about 53% of them did show NIFH amplification. So it seems like these genes are pretty well distributed, suggesting that nitrogen fixation might be occurring. And in this one sample I just pointed out at 3,500 meters water depth, we sequenced the NIFH genes and found that they're mostly hosted by delta proteobacteria. So in this table, you can see the number of NIFH ASVs that fell into each one of these categories. We were able to identify a host for the NIFH genes using software that we're calling PPIT that was recently developed by one of my graduate students. So you can look up that paper here if you're interested in how we did that. But the summary is, it seems like delta proteobacteria, which we know are very abundant throughout the transect, are capable of nitrogen fixation. So are these sediments actually nitrogen or carbon limited? We did a direct experiment to test that. So this is the amount of ammonium assimilated at each of these three water depths indicated up here. Each of the colors of these dots indicates a different depth horizon in the sediments. And so these are the, this is the total anabolic activity we saw. We also did an experiment where we added unlabeled glucose to see if we could stimulate activity by adding a source, an easily digestible source of carbon. And what we saw was that we could increase activity. This was a statistically significant increase by adding carbon. So this does suggest carbon limitation. When we did a similar experiment in reverse, so now we're looking at 13C glucose uptake, which is our proxy for heterotrophy, and we added ammonium to see if we could stimulate the glucose uptake. We did not see a statistically significant increase in uptake. So what we see here is evidence for carbon limitation, but not nitrogen limitation, despite those low concentrations of ammonium. We looked for nitrogen fixation directly at all 5 sites. And as you can see here, we saw no evidence for nitrogen fixation despite the genes being present. So what this is really telling us, in combination with the previous slide that I just told you, is that although nitrogen concentrations may be low, especially in the deep surface samples, it's not low enough to stimulate nitrogen fixation. The genetic capability does seem to be there though. So what we think might be happening is in different situations on long time scales. For instance, a methane seep opens up, a whale falls. The community is capable of fixing nitrogen if it's driven to the ratios of carbon and nitrogen that would make that necessary, which is kind of an interesting safety net they have for the future. So we also looked at the concentrate or the uptake of inorganic carbon, so bicarbonate or autotrophy. What we can see there is we see significantly higher uptake of inorganic carbon than nitrogen fixation, which is another indication that really carbon is more limiting than nitrogen. So what is controlling this distribution of activity? We correlated our rates with depth in the sediment. This is all data just from the West Coast transect, as well as distance from shore and chlorophyll and several other parameters, but these are the 3 that gave us statistically significant correlations. And we did the same thing at our other site. And the reason it's really nice to have this other site is because we have a really different distance from coast depth relationship on the East Coast of the US. It gets deeper, basically much further away from shore. And when we look at it here, we're adding this data to the previous data, we can see that we no longer have a statistically significant relationship with depth, which also indicates that pressure is not the primary controlling factor here. So we conducted all of our experiments at 2 times atmospheric pressure, which is not close to what they were experiencing of easily 100 times atmospheric pressure. So the fact that we did not see a significant correlation here means that our results are not just an effect of variable kind of in optimization to the particular conditions that we tested. We did see a statistically significant trend with distance from land and chlorophyll, even more so when we added these other samples and a few previously published rates. So these are the 2 parameters that we really continued to look into. So using distance from land as our predictor of microbial activity, we can actually calculate a global rate because we can calculate the area of the seafloor, essentially, that is a particular distance from land around the world. And we can therefore extrapolate to a global estimate of benthic activity. We did the same thing for the chlorophyll concentrations. And sorry, I didn't exactly explain what this was. This is chlorophyll concentration in the overlying water. So no matter what the depth is, what the overlying chlorophyll concentration is. This is also something that's known worldwide because of satellite data, so that we could calculate a global estimate using this predictor as well. And we get pretty similar numbers compared to the global estimates that have been predicted before. These are within the range, but probably on the lower side of what's been seen before. The really interesting thing about doing it this way, so looking at the distance from coast, is that we can actually see how that might have changed through time. So if we use data that predicts basically how the position of the continents have changed in the last 400 million years, and we predict the amount of activity that would have changed, everything else being constant, given those different configurations, you can see that when there was essentially less coastline, there would be less microbial activity in the sediments. And so this basically amounts to a 40% difference due only to the position of the continents over time. And there's a lot of assumptions that go into this. We could go into that in more detail, but I think it's interesting to think that microbial activity and tectonic activity could be linked kind of in such a significant way. Whether this has specific effects on carbon burial. So the percentage of the fixed carbon that's buried really depends on how photosynthetic activity changes at the same time that these changes are happening. If they change together, then it just does not change the ratio and we don't have implications for atmospheric O2 or redox. But if they change asynchronously, then maybe it would make a difference. So that would be something that would be interesting to look into in future work. So to wrap up this first section, we see that activity decreases with distance from shore and depth in sediment. And I didn't explain it in detail, but we saw high replicability at the 1 meter scale. So we had spatial replicates at 1 meters. We saw that organic carbon seems to be the primary control on activity, whether that's because it's a source of carbon, electrons, or energy, we can't parse out, but this seems to be the main controller. And that is, of course, related to distance from land. That allows us to roughly estimate both the total activity on Earth today and how it might have changed, at least relatively, over time. And it would be interesting to look into how that changed relative to photosynthetic carbon fixation to try to get an idea of how carbon burial might have changed over time. So moving on to our second story, let me take a look at the time. All right, microbial activity and metabolic capabilities in the dark ocean. So now I'm going to talk a little bit more about nanoSIMS data, which I think is something that many of you are familiar with, but I will just summarize quickly again. In this approach, we use a primary ion beam with a very small radius or diameter of about 50 nanometers, which gives us a spatial resolution of about that much. We can raster this primary beam across our sample like an old-fashioned TV and actually get a map of the ions in that sample. So here's just the schematic. You put your sample in a vacuum here, kind of like you would prepare it for an SEM. Your primary beam hits your sample, generates secondary ions. These are separated, and you can measure up to 7 masses at once. So as I showed on my title slide, this is an example of a filter with marine microbial cells as seen through the nanoSIMS. So each one of these are about 2 micron cells, and we don't know who they are, but we can get the isotopic composition of each one of them individually in this manner. Previous nanoSIMS studies have often spent a lot of time focusing on particular interactions between identified cells. And the next story I'm going to talk about does that quite a bit. Another approach you can take with the nanoSIMS that hasn't been done quite as much, but I'm getting more and more interested in, is this kind of scanning mode. So you take a picture of the entire community and you don't know who the cells are, but you get an idea of the variability in total activity in the community as a whole. And depending on which substrates you add and in what combination, you can actually rapidly screen if they're autotrophic, heterotrophic, et cetera, and get percentages of these kind of major metabolic groups in the community as a whole, which I think is pretty neat. So my postdoc, Nestor Arandia-Gorostidi, is one of the people taking this approach in my lab. So he collected seawater off the west coast of the US, so in the Pacific Ocean, from 50 meters to a little over 3,000 meters water depth. And he added amino acids, 15 N amino acids, ammonium, and urea. And you can see that the incorporation of these substrates decreases with depth. But what you're missing in kind of a bulk plot like this is how the variability in uptake within the community changes. And that's what we can get with the single cell approach. So for each one of these substrates, now I'm showing how enriched these cells are. So more enriched is to the right on this axis. And if it's green, it means it was enriched. And how that distribution of cells looked like for these different substrates. You can see in the surface sample for urea, only about 8% of the cells total consumed this 15N urea, but for ammonium and amino acids, we're closer to 90%. So this is telling us both that the vast majority of cells in the surface of the ocean are anabolically active, over 90% even in this short incubation, and that some are also capable, some but fewer are capable of urea assimilation. You also see really different patterns of distribution between the ammonium and amino acid uptake, and we're working on ways to quantify that difference so that we can compare that kind of broadly through different substrates and different depths. You can now compare this data to all of the samples with depth and see some pretty interesting trends. The ammonium and amino acids follow a similar trend in which fewer and fewer cells incorporate this substrates with depth, although still the majority of the cells, over 50%, are active throughout the water column. And something really interesting happens with urea, where you have a major peak in the percentage of cells that can consume it at about 150 meters. You have almost 75% of the cells assimilating it, whereas you have so many fewer above and below. Another thing to note here is the higher percentage of cells, relatively speaking, that rely on urea relative to these other substrates with depth. In the surface, it's really a minority of the cells that are capable of assimilating it. But at depth, you have over half of the cells that are assimilating these other substrates capable of assimilating urea. So we think that urea is a more significant substrate with depth than it is at the surface. So let's get back to this one depth where we have a much higher percentage of cells assimilating urea. When we look at the 16S data from this same water transect, we see that something else interesting happens at 150 meters, and that is we see a big spike in Thaumarchaeota relative to the surface sample. So this is where the Thaumarchaeota become abundant, and they maintain abundant throughout the rest of the water column, but this is likely where they are the most active. So in combination with other work that has shown that Thaumarchaeota are able to use urea as a substrate for nitrification, we are suspecting that it is Thaumarchaeota that are responsible for most of this assimilation. So let's take a step back and think a little bit more about the Thaumarchaeota for a minute. They are an abundant and enigmatic deep sea phylum. I say phylum here, but they're likely going to be reclassified. I won't get into that whole story right now. At the surface of the water column, they only make up about 1% of the total cells, but as you go deep into the water, so here's this entire slide is a little graphic of the ocean, they become more and more abundant until they're as abundant as bacteria or even more abundant with depth, which is kind of alarming. You might not have even heard of this group before, and now you're hearing that they're more abundant than total bacteria with depth in the ocean. That means that altogether, archaea comprise about 30% of all of the cells in the ocean, and 80% of those archaea are these Thaumarchaeota. They're previously called the marine group 1 Crenarchaeota, and as I mentioned, in the future, they might be reclassified as the Nitrosphaeria, but we're kind of waiting a final stamp of approval on that reclassification. So for now, I'll be calling them the Thaumarchaeota or marine group 1. So for a long time, it was unknown what these organisms did in general. About 15 years ago now, the first isolate demonstrated that they were capable of ammonia oxidation, aerobic ammonia oxidation, which was significant because that is a chemoautotrophic process. They're fixing inorganic carbon to couple with that ammonia oxidation. So this is basically a source of bioavailable carbon deep in the water column. As they perform this metabolism, they also produce nitrous oxide, which is a really potent greenhouse gas. So we care about them for the cycling of both nitrous oxide and CO2. But one of the big questions about them has remained, are they capable of taking up organic carbon or nitrogen? As I mentioned, there's already some interesting things known about urea and their ability to use urea as a source, as a substrate for nitrification. So there's a lot of evidence kind of on both sides of whether or not they use organic or inorganic carbon from pure cultures. There are limited pure cultures available of this group, mostly representatives from the shallow water, much less so from the more abundant deep clades. There's genomic evidence that shows they have the genetic capability to use inorganic carbon, the 3-hydroxypropionate 4-hydroxybutyrate pathway in particular, and stable isotope tracing that has shown the direct uptake of 13C bicarbonate. But there's also a lot of evidence that they can take up organic molecules. So that also comes from genetic work. They have the oxidative TCA cycle in some cases, transporters for organic matter. And there's been specific identification of the uptake of tritiated amino acids, some but not many examples of 13C uptake, and this particular method and this particular site is a little bit, it's questionable how representative that was of open waters. But maybe the most important study in this respect was one that came out about 15 years ago using radiocarbon that showed that about 20% of deep Thaumarchaeota carbon was linked to organic matter. So it was unknown at that point if all of the Thaumarchaeota, each individual cell, was getting about 20% of their carbon from organic sources, or if there was a split in the phylum where some of them or most of them were autotrophic and then about 20% were totally heterotrophic. That bulk analysis couldn't differentiate between those two possibilities. So the potential explanations for these conflicting observations, I'll just kind of summarize graphically because it can be a little confusing, is that, like I said, we have these two groups within the phylum that are partially autotrophs and partially heterotrophs. You could also have all mixotrophs. You could have some combination of the three, autotrophs, mixotrophs, and heterotrophs, or you could have mostly autotrophs, some of which also take up some organic matter. So this is a question that we can solve using a single cell approach, because we can actually look at the percentages of carbon that each individual cell is getting from different sources. So to answer this, we went out into the Pacific Ocean, just 17 kilometers off the coast of LA. We didn't have to do a big cruise expedition for this. Like I said, these organisms are so abundant, we did not have to go far. We collected our samples at 150 meters water depth. We did metagenomics and metatranscriptomics and 13C and 15N incubations. And I'll be talking primarily about the latter, but I will first tell you that in our omics work, we did identify Thaumarchaeota mags that contained both organic and inorganic pathways that I mentioned before have been identified. And even in particular mags, they were transcribing the genes involved in both simultaneously, as if they were capable of taking up both organic and inorganic carbon at once. So to directly see if this was the case, we incubated with a whole suite of isotopically labeled substrates. We picked these substrates primarily because they're intermediates in the metabolic pathways for which we saw genetic evidence, and also because they are frequently assimilated by many marine organisms like amino acids, protein, and I already told you we had a specific hypothesis for urea. We identified these cells using fluorescence in situ hybridization before the nanoSIMS analysis, and then we were able to say specifically for this group what their metabolic capabilities were. So for those of you unfamiliar with fish, this is where we can add fluorescently labeled oligonucleotide probes and actually physically turn the cell a particular color. We can find then the Thaumarchaeota within the mixed samples and analyze that exact cell in the nanoSIMS to get its isotopic composition. So different approach from that overall scanning one I mentioned before. So can the marine group 1 Thaumarchaeota assimilate carbon? To orient you on this graph, we're looking at the net assimilation of carbon of these different substrates here. Right now, I'm only showing you bacteria in purple and DAPI in gray. So DAPI is just going to be a combination of the bacteria and archaeal cells that we did not differentiate. These are the number of cells that are enriched out of the total that we analyzed down here. So when we analyzed the Thaumarchaeota, we saw that they did not take up glucose pyruvate or oxaloacetate. And when we looked at urea and amino acids, we only saw the tiniest bit of incorporation, but it was incorporation nonetheless in these two substrates. So the difference between urea and amino acids in these three substrates is that they are nitrogenous, so they contain nitrogen. And that really seems to be a theme in their organic matter utilization. They were not interested in any of the purely organic carbon substrates, but if it contained nitrogen, they did assimilate some of the carbon from that molecule. We also looked at the 15N assimilation from ammonium, urea, amino acids, and protein. And what we see here is something quite different. So we see a lot of nitrogen incorporation from urea and amino acids, the same as we saw from ammonium, actually, showing that organic nitrogen is potentially as significant of a source of nitrogen to them as ammonium, but none from protein. We did not see any 13C incorporation from protein either. So putting this together, how much does it matter? What percent of the Mg1 Thaumarchaeota carbon is organic? Well, we can see just from the single cell analysis that 15% of the cells assimilate carbon from urea and 36% of the cells assimilate carbon from amino acids. But that doesn't tell you how significant of a carbon source it is for those cells. So to answer that, we actually took the ratio of the net carbon assimilation to the net nitrogen assimilation to calculate that of the cells assimilating urea, it made up to 5% of their total carbon diet. So this was not a primary carbon source. Amino acids contributed more carbon, about 14% of the total, but again, not the primary carbon source. So we can put that together then and say that for the entire population, about 0.5 to 11% of their carbon diet is derived from this nitrogenous organic matter. So to summarize, the metagenomics and metatranscriptomics data showed us that they do appear to have the genetic capability for both organic and inorganic carbon assimilation, which is what has led people to hypothesize that there is such extensive mixotrophy in this group. But our nanoSIMS data really showed a different story, which is that they're not taking up extensive organic matter. What we think this means is a lot of those genes involved in organic matter that are detected and frequently kind of misinterpreted, they're actually genes involved or enzymes involved in internal processing of organic matter, because all organisms, whether they're autotrophic or heterotrophic, have to do certain internal processing of organic matter. And that doesn't mean that they're specifically taking exogenous organic matter and using that into their biomass. But we do see incorporation from urea and amino acids, and it does make up a small percentage of their total carbon budget. So in summary, we think that they are really primarily autotrophs and mixotrophs. And for the ones that are mixotrophs, they're really all about the nitrogen. This probably relates back to their need for nitrogen for nitrification, particularly in the case of urea. If they can cleave urea, they can oxidize that ammonia and also take up the carbon and nitrogen once it's in the cell. So it's probably related to their catabolic needs. So I have a few more minutes to discuss my third topic. Neha, would you like me to wrap up now for questions, or would you like me to discuss this third?
I think it's okay. You can discuss the third and then we can open up for questions. We have some.
Okay, I'll try to keep it just for 5 minutes so we can still have some time.
Okay.
Okay. So in this final little story, I'm going to talk about work that was led by my postdoc, Isabel Klawan, who did really beautiful work with diatom fungal parasitism. So she actually worked with a system that is a model system that was enriched from lake samples, but is really relevant for the marine photic zone. So as many of you know, phototrophs are an extremely important part of our world's biogeochemical cycles, and in particular, single cell phytoplankton contribute almost half of the world's primary production. You might not know that fungal parasites are actually extremely common. And when we think about aquatic food webs, they're usually not kind of at the center of the discussion. But what we are discussing is the carbon that directly flows from these diatoms into the fungus, which we're calling a fungal shunt, kind of similar to the viral shunt in aquatic food webs. And we're looking at exactly how big that effect is and how that affects heterotrophic bacteria. So like I said, this is a model system. She grew diatoms and split the culture into unaffected and affected, infected. So basically adding this particular fungal parasite. And she quantified what the effect was on heterotrophic colonization of these diatoms, found that diatoms were 2 to 4 times more heavily colonized by bacteria when they were infected by the fungi, and it significantly changed the composition of those attached bacteria as well. When we analyzed these diatoms, we actually used 2 different SIMS instruments, one that has a stronger primary ion beam in addition to the nanoSIMS, which has a less strong ion beam, higher spatial resolution, which helped us analyze kind of into the thick frustule of the diatom. So we were able to get isotopic compositions of both the diatoms, the fungi, and the heterotrophic bacteria. When we did this, we were able to compare the carbon, the 13C carbon composition, which was completely from the 13C bicarbonate we added. We could see that 100% of the carbon in the fungal sporangia was derived from the diatom, and that was the same as the zoospores that the fungi eventually open up into in that stage of their life cycle. The associated bacteria derives less of their carbon from the diatom, and the free-living bacteria even less. And you can see that kind of as you go down here, and these groups become less and less enriched to the left. So if you use those results and model them to rates of fungal occurrences in a natural lake, you can see in this pink wedge here, this is the percentage of photosynthetic C in the fungal fraction, and you can see that comprises 20% of all of the photosynthates at the peak of infection, indicating that this is really a significant amount of carbon that ends up being shuttled through the fungi instead of kind of directly being either kept into the diatom or leaked out and going through the microbial loop, which is probably the fate of that carbon otherwise. So kind of to summarize that, where we usually think of photosynthates in a diatom being transferred either to DOC, which can then go through the microbial loop or be circled back to DOC by the viral shunt. The fungal shunt is basically taking carbon from the diatom, which then, when the sporangium then opens up into zoospores, these can be directly consumed by higher trophic levels. So it's basically a way to shuttle this carbon directly from the diatom into higher trophic levels and basically bypass the microbial loop. So this is definitely a significant pathway that should be considered in aquatic food webs. And not just because of the magnitude of the directional change of the carbon, but also we're seeing community shifts in the microbes. Not surprisingly, when this shift is involved, different organisms are attaching to the diatoms, and it's changing the composition both of the attached bacteria and the free-living community as well. So the overall summary from the first part of my talk, if we can even think back that far, is that benthic microbial activity is limited by organic carbon, and activity may have varied by at least 40% in the last 400 million years based on tectonic activity. We see activity in the dark water column, and particularly the Thaumarchaeota, really high percentages of active cells, which we can see with our really sensitive nanoSIMS assessment. And we can also conclude that Thaumarchaeota derive a small fraction overall of their biomass from carbon and organic matter, but maybe most interestingly, this entire fraction was coming from nitrogenous organic material. So it changes how we think they're interacting with the types of organic material around them. We saw that fungal diatom parasitism can shunt significant percentages of this photosynthetic carbon in aquatic food webs. And the overall kind of approach here that we're seeing with all of the different stories I've told is that quantifying the activity and identifying controls facilitates extrapolations in space and time, which hopefully we can link to biogeochemical models and really link this single cell activity to larger scale processes. And I would like to thank all of my group members, particularly Nicolette Meyer, Nesta Arandia Gorestivi, Isabel Klawoon, and Alma Parada, as well as other group members for their contribution to all of this work. So thank you very much, and I am happy to answer any questions you have.
Thanks, Anne, for a wonderful talk. If anyone have a question, either they can raise their hand or type in the chat, or just, I think I can see everyone pretty much. I see a question from Kareem.
Yes. So thank you, and that was really nice. And I guess I noted down again many questions, but I want to confiscate all the time, but maybe ask you later on a few of them. So two questions maybe. So the first one is on the first part, you mentioned the carbon versus nitrogen limitation, or testing the carbon versus nitrogen limitation. I wanted to ask, what about phosphorus? I guess that might be complicated to test maybe, but so that's a usual suspect as well in this environment. So I was wondering if you had any idea about, I don't know, maybe in the sediments you have high orthophosphate concentration in solution. I don't know.
That's a really great question. And we can look into that because we have phosphate concentrations. I am assuming that there was not a significant correlation with phosphate concentrations, and that's why I didn't have a slide on it, but I should revisit that more closely. And we didn't do any particular manipulation experiments to test it directly, but I could at least revisit the pore water concentrations to look for a correlative relationship.
Okay, thanks. And if I may, so jumping to the third section, so do you have any idea about how much of these fungal shunt will eventually, I mean, what it becomes afterwards? Does it settle down, sediment to the bottom, and how? Because I guess so. I guess my view so far was like you have huge biomass in the diatoms, and from what I understood is that in the models, diatoms are maybe you need first to aggregate them, but this is a pretty efficient sink because it can sediment settle down pretty easily. So I guess if you start to shunt 20% of it to other organisms, I have no clue whether they might sediment as well, whether you will find a big proportion in the sediments or not.
Yeah, our current understanding, which is really evolving, I think we don't know enough about how fungi are affecting these systems, is that when the sporangia eventually go into their next life stage, which is to release these zoospores, they are consumed by higher trophic levels. So it's basically going into zooplankton and other larger organisms to whatever fate they end up having. Usually we think of carbon going up the food chain as a way to aggregate it and ultimately bury more. So is that more or less efficient than it being in the diatom and aggregating and sinking that way? I don't know yet. So I think that there's a lot of work that needs to be done to figure that out. I do think, though, that as an alternative to the photosynthetic carbon being released as DOC and then being kind of recycled in the microbial loop, which results in a lot of respiration and return to CO2, my guess might be that this is actually a more efficient way to bury carbon, but I don't have enough kind of experimental evidence to support that to say for sure.
Okay, any other questions? Well, people may be thinking about questions. I have one quick question, maybe a little more broader. I think it comes from when you summarize the talk about how, and I guess you've also pointed out in the motivation about how the activity in the deep sea is limited by the organic carbon. And I'm just wondering, have you, what's your perspective on this whole idea of like how the types of organic or the carbon which is available anymore in the deep sea is changing? And it's given the anthropogenic inputs and how less labile these, or even already we know the carbon in the deep sea is way more less labile, but what's the perspective on when you couple that together, the changing nature of the organic carbon being buried, and how does that change the whole story?
Yes, this is a really great question. And I wish there was a cheaper, easier way to assess the reactivity and the composition of organic matter, because it would be a way easier way to incorporate that into the story. It's unsatisfying to me to just say organic carbon as if it's one thing, but it's where we are right now. And I think that future work could really dig into that in much more detail and figure out how it is degrading in the sediments and how specifically that affects the activity. And how we see that reflected too in the metagenomics, where we see the capabilities of these organisms to degrade different types of organic carbon. So you're onto something really important there. And I'm looking forward to getting more into it is basically all I can say right now.
Okay. Okay. Any other questions? I think I'm gonna give the floor to Karim since he has another question, I suppose.
Yeah, I mean, literally, I have several ones, but so yeah. So another one. About the Thaumarchaeota. Did I miss it? Did you test the bicarbonate incorporation?
Yes, I didn't show that work.
Okay.
But yes, they are taking up the bicarbonate. We see in over 90% of the cells are taking up bicarbonate. Okay.
And so the question is, because these are, I mean, really interesting experiment. I mean, very, very powerful experiment to assess the metabolism of those of microbes we don't know. So I have two questions about that. So practically, you just spike, right, the solution, the sample. I mean, so you already have the bicarbonate and anything else from your sample, and you just add the label bicarbonate, right?
Exactly. Since there's so much bicarbonate in marine water already, we end up with a relatively low percentage label. So we add a ton of 13C bicarbonate, but it's still only 10 or 20% labeled total. So our detection limit for uptake of bicarbonate is actually way lower than it is for the organic substrates, because we can add those and have them be 90, 95% labeled in the total pool. So we are limited by how much bicarbonate there is in the sample to begin with, if that answers your question.
Yeah, and so related one, and again, this is pretty specific, but do you see any, so when you show the nitrogen enrichment, for example, do you see any connection or relationship between the enrichment and how high is the enrichment and the size of the organisms?
Specifically in the Thaumarchaeota or any of the cells?
Any, I guess, any, because you had also the first, I mean, yeah.
You know, I don't think we've done that, but we definitely should. That would be very interesting to know, because it would be nice if size could be a proxy for activity or growth, really roughly. So we should definitely look at that. That would be easy to do. Thanks for the suggestion.
And the last thing, and sorry, there's a small short one, but in the first part, so I understand this is bulk measurements, but I was wondering, so you're in the sediments, so I could see that you use butyl bottles. So and so the headspace, what is the headspace? Do you preserve, like, do you keep the anoxic conditions when you have anoxic samples, or do you don't care about that?
So in the sediment incubations, we definitely keep them anoxic, and we did oxygen pore water measurements and found that the sediments were anoxic after the first few millimeters. So we did all of the sediments completely anoxic. That means we might have underestimated any aerobic activity in those first few millimeters in our 0 to 5 sample. So that's an underestimate. But yeah, they were anaerobic. Or anoxic. In the water column ones, we do not keep them anoxic. It is oxygenated waters, but I have thought about how just keeping them in these polycarbonate bottles might end up having different oxygen concentrations than the in situ environment. And that's one of the things we should think about when kind of extrapolating our rates to those other environments. But I don't think it should affect whether or not they were taking up organic matter necessarily, but it's something to think about.
Thanks.
Francois, you have a question?
Yeah, thank you very much, and thank you for the great talk. It's very, very interesting. I just because I don't know at all about these methods. From a methodological point of view, for instance, if we take the labeled bicarbonate, for instance, are there cases where these could be somewhat concentrated at the single cell level and having nothing to do with the metabolism of the cell? And conversely, I would say, are there some example of strong reactivity of labeled substrates that would be diluted then? Well, it's just to know about that literature and about that field.
Yeah.
What are the main maybe methodological issues? And the other question was, I was not sure if so that was the first one. It's really about a non-metabolic fate of our exchange between the cells, as you would do on adsorption on minerals, for instance, or things like that. And the other things is at some point you have to measure precisely the ratio of the enrichment to get that percentages. I mean, or because you have one cell that has, for instance, been enriched in N15, or you have to measure that precisely, the ratio N15 over N14 in that cell. And in that case, if you do, is there an analytical difficulty, especially with the nanoSIMS, in order to get a kind of quantitative ratio at some point?
So there's a lot there.
Yeah,
so
I'm sorry. Because I don't know the method, so I'm just wondering.
Yes, great questions. I'm not going to be able to do all of them justice, but I'll give you at least a little bit. So with respect to any kind of abiotic attachment of these substrates to the cells, we measure all of the uptake rates relative to T0 cells that have had all of the substrates added and then were filtered immediately. So if we assume that that abiotic stickiness would happen immediately, then we're taking that into account when we're looking at the difference between the T0 and the T final assessment. The 15N ammonium does stick to clay particles in the sediments a lot. So that's actually one of the big caveats of bulk sediment isotopic measurements. It's much harder than a nanoSIMS measurement where you're looking at a relatively clean cell when you have the sediment included in the measurement. So we do a lot of chemical washes of the sediment to try to release that 15N from the clays, which generally works. It's a KCl potassium chloride rinse. And so we've done a bunch of controls where we try to get a baseline idea of what that abiotic attachment is. And again, that's what we compare our enrichment to when we're calculating rates. In terms of other biological processes that can result in like an erroneous conclusion of metabolic activity, there's two things that come to mind. One is what we call cross-feeding or recycling of substrates. So if one organism consumes it and then excretes it as a different molecule and then a second one does. For measurements of total activity, we actually don't care if that happens because it doesn't matter if that's specific molecule. We just want to know if anybody has taken up anything. For the measurements where we're trying to quantify the particular metabolisms, that's definitely a big concern. And it's a particular concern in situations where the background concentration of that substrate is low. So in the case of adding 13C organic matter to an environment where there isn't very much organic matter, any product that's 13C labeled could then be consumed by the rest of the community and appear that it has been assimilated. The way that we try to differentiate between primary consumption and secondary consumption is to use two different labels. So if we add the 13C organic matter as the primary thing we're interested in, but we also add 15N ammonium as the measure of total anabolic activity, we can see if the 13C incorporation is stoichiometrically significant compared to the total 15N. So basically, if it's a big percentage of that what is assimilated versus a small percentage and get an idea of if it was primary or secondary. We can't always differentiate. So there's some potential possibility that it was always recycled from something else. In the case of substrates that are extremely abundant in the environment, like bicarbonate, it's very unlikely that something would receive it because it would be so diluted in that pool. So if you were concerned that an autotroph would end up 13C enriched because we added 13C organic matter to the system, like in the Thaumarchaeota case, we actually don't really need to worry about that, because if the 13C, for instance, glucose was converted to 13C bicarbonate in the marine sample, it wouldn't change the background isotopic composition of the bicarbonate enough to show up as the 13C enrichment in the autotroph. We've done that calculation, and we even measured the isotopic composition of the bicarbonate to make sure that that could not be what we're seeing in our Thaumarchaeota. So I guess the short answer is there are a lot of caveats and concerns with this experimental design, but we try to get around them by using secondary isotopes and thinking about the whole system. And certain questions we can answer well, and other questions we can't really because of these problems. And so we pick that way.
Thank you very much.
Sure.
Any other questions? Well, I think we are running out of time anyways, but we'll take one last question from Robin Havas, if I pronounce the name right.
Yes, thank you. I don't know if you can hear me. Yeah, okay. Thank you for the talk, for a great talk. I was just curious to know, you mentioned earlier, you estimated about 20% of diatom fixed carbon that was like shunt through this fungi thing. And I was just curious, I wanted to ask you if you had an estimation of the amount of fixed carbon that was released as DOC, either from diatoms or from fungi.
So the effect of infection on the DOC release? This was actually a very complicated part of the experiment, because we saw that the amount of DOC did not increase with infection, but we saw that the 13C content of the DOC did increase. So something happened with the type of DOC that was comprising the pool or what was being consumed. We haven't fully worked that out yet.
Okay, so the type of DOC that was released was maybe different according to the infection you added.
Right.
Okay.
Right.
And so I'm not sure I got that right, but you said you had this from lacustrine environments. Did you have what were the concentrations of these relative pools of carbon in those lacustrine environments? So like concentration of diatoms and relative to DOC, for example, and size-wise compared to the fungi infection?
I don't remember those numbers.
Okay.
Yeah, you could either email me and I could look it up, or this paper is in press now, so it should be coming out soon.
Okay.
But yeah, I can follow up if you'd like.
Okay, thank you.
Sorry about that.
No problem. Thank you very much.
Okay. Well, if there are no last minute questions, I would like to thank Anne for a very good talk, and really, I think it was really fascinating to see such a diversity in our marine environments. So it was really cool. So thank you so much, and thank you everyone for joining us. And we look forward to seeing you all for our next seminar. Thank you.
Thank you very much.
Thank you very much.
Bye.